Okay, can people online hear us? Have we checked that yet? Or we are okay? Um, okay, I will just get started on this. Thank you very much for this opportunity. And yes, people can hear. Thank you so much for saying that. And this is my great pleasure to introduce today's speaker, Dr. Xu Dong. And Dr. Xu is a leading scientist in bioinformatics and computational biology with a distinguished career spanning academia and actually national laboratories. He's currently a professor at the University of South Florida. When we invited him, he was actually at the University of Missouri, where he served as the curator's distinguished professor and the PI of the digital biology laboratory. And he received his PhD from the University of Illinois at Urbana-Champaign in 1995 and completed postdoc training at the US National Cancer Institute and worked as a staff scientist at the Oak Ridge National Laboratory before transitioning back into academia. At Missouri, he held key leadership roles, including 10 years as the chair of the Department of Computer Sciences from 2017 to 2016, and the director of the Information Technology Program from 2017 to 2020. Past 30 years, Dr. Xu has made broad and impactful contributions to computational biology domain, including work in single-cell data analysis, protein structure prediction, protein localization, and in general, computational systems biology. And in the past 10 years, his research has been mainly focused on integrating deep learning with bioinformatics in the areas such as protein language models and connecting genotype to phenotypes. And he has over 500 publications in his career and more than 31,000 citations and the H index of 92. And he's also elected as fellow of both the AAAS, American Association for the Advancement of Sciences, and the American Institute for Medical and Biological Engineering. So this highlights his achievement across both sciences and so with this, it's really my great pleasure to introduce Dr. Xu Dong, and please join me welcome Dr. Xu, who will share his insight about cutting-edge approaches using deep learning in single-cell research. Please go ahead.
All right. Yeah, thanks, Tom, for the kind introduction. Yeah, I also like to thank the organizer for inviting me here. It's a great pleasure to share some of our research in deep learning methods for single-cell data analysis. Yeah, so uh, when you analyze the hundreds of tools you can use, you don't have to use deep learning methods. There are many good statistical methods and other computer science methods. But there is some unique aspect for single cell that could be beneficial to use deep learning methods. First of all, the single cell data size is huge. We're talking about from hundreds to millions of cells from one measurement. And then because of the size of data, it naturally fits to deep learning because most deep learning methods are so-called data hungry algorithms. So big data definitely help. Another issue single cell data often face is so-called sparsity. That is the high dropout rate and multiple noises in all kinds of single cell data. So that's why over the past about 5-6 years, we have been focusing on applying deep learning methods for single cell data analysis. We actually explored various aspects of machine learning methods for all kinds of modality or aspects of a single cell data analysis. My longtime collaborator, Dr. Qingma, and I wrote a couple of reviews in Nature Review of Molecular Cell Biology. So if you'd like to know the broad aspect of this direction, you can read our papers. But today, I like to just focus on some of our own applications. You can see from these use cases why deep learning can play an important role to mine more accurate data, information, to build better hypotheses and models. So I will use 3 examples. The first example is to use graph neural networks for single cell data analysis. The second one is that how to prompt large single-cell RNA-seq models. The third one is that large single-cell model for plants. So let's see the graph neural network aspect. So this work we started 2021. So at that time, actually most methods just use simple graph methods for single-cell analysis. That is, you can build like a gene by cell matrix from single-cell RNA-seq data. And then you can also build graph or use other methods to do this analysis. But we found that by using graph neural network, you can really get results more accurately. So in this case, we first do embedding, embedding from to build cell representation using genes. So here is each cell, we have, let's say, 20-some thousand genes. But here you have each cell represented by a vector through this autoencoder. And then we actually use this graph structure. That is, if two cells are similar in the representation, then we build an edge. Then we use the so-called graph autoencoder to build graph representation. And then we do clustering for each cluster. Then we do auto encoder again. Then we go here and then we do this iteratively. Through a few iterations, we can build much better representation that allows us to divide cells into different clusters or to identify cell-cell relationships. And then we can also do imputation a lot better. So through this approach, it really opens the door for using graph network for single-cell data analysis, because we were actually pretty much the first method, graph neural net method for single-cell data analysis. A lot of people actually followed our method. That's why our paper was cited for more than 770 times. So I just want to explain why graph network can help. So if you think like a cell by gene matrix, you see that there are a lot of missing values and a lot of noises. You can do a simple average. But by doing a simple average, you lost so-called this cell heterogeneity. Even you resolve the signal to noise issue by averaging, but you won't be able to see different cell groups, what kind of features they have. But graph neural network actually does this in a little bit different way. That is, you have this graph structure. So here, each node represents a cell. Then you can pretty much use the neighbors of your cell to build a mini cohort. But again, this is not a simple average. You use the AI to make it a smarter integration. That means depending on the connection, you give different weights. So you can basically do this averaging through this mini cohort. And then you can keep iterating this process. That means that each cell you will use its neighbor information, and then you can gradually propagate to the entire network. So that's why through this smart aggregation, your signal to noise ratio is much better. You can get also a lot of heterogeneity information from this graph network approach. And then we extended this method to the spatial transcriptome data. So here, it is even easier to build a graph because naturally two neighboring cells in 3D space or in 2D space can build an edge. And then we can also do this graph autoencoder. And then what we did is actually to project the embedding into three-dimensional space corresponding to RGB, a red, green, blue color scheme. So you can visualize the image. And then you can use the image segmentation techniques to actually divide cells into different cell architectures. And then by doing that, we really see the cell architecture in details. So first of all, these architectures through RGB imaging is actually closely related to tissue architecture. Second of all, we see many details from such visualization. And then we actually worked with clinicians to annotate some pathology data. So here is actually the spatial transcriptome data for cancer tissues. And then the pathologists actually used our initial segmentation and then to see whether it corresponds to meaningful cell type annotations. And they found actually the mapping is very clear. That is, for example, in this group, it actually corresponds to good blood cells. This one corresponds to tumor cells, etc. So it really maps to pathology annotation very well. And then we further expand the graph approach to integrate different modality of single cell data. So here, the input would be two kinds of or multiple kinds of data. For example, you can have two RNA-seq data from two different labs and how to integrate them by addressing batch effect, etc. And then you can have a protein RNA data, you can have a RNA ATAC data. And then we use this so-called heterogeneous graph transformer architecture to integrate this data. So here, the reason why we call it a heterogeneous graph is that each node could represent either a cell or a gene. Then we can use this message passing to connect them. And then through this graph representation, we can build a cell-gene graph, cell graph, gene-gene graph, etc. And then we can eventually resolve cell type specific gene networks. And then we can do all kinds of downstream tasks like a cell type annotation, markers, network, etc. So here is how we do it. Let me just use an example of RNA-seq data and ATAC data, how we integrate them. So RNA-seq data is pretty straightforward. So you can easily build so-called gene by cell matrix for RNA-seq data. And then ATAC data, you have the peaks for the regulatory regions. So we need to do some operation. First of all, to filter some non-informative peaks, and then we calculate some regulatory potentials. That means if the peak is closer to the gene start site, it's actually more informative, so give a higher weight. And then combined, we can aggregate all the peaks for the upstream region of a single gene. Then we combine all these effects, and then we can also construct gene by cell matrix for the so-called regulatory potential. And then we integrate both sets of data into the graph transformer. That would allow us to build cell type specific regulatory network directly. So that's the so-called end-to-end approach. Instead of you identify cell type first, then you use a separate tool to build network. So we found that to do this end-to-end approach is actually more informative and more effective. So here shows the network we build for different cell types. For example, this is CD8 T cells, this is memory T cells. You see that different cell types, they really have quite different regulatory networks that are active for this cell type. So this, of course, is still a very challenging problem, but that at least opened a door to build hypotheses for cell type specific network. So another problem we use the graph network is to identify so-called network motif. So here is a spatial transcriptome data. Each dot represent one cell, each color represent one cell type. Then you see it actually forms some pattern. Um, Sometimes people call it a cellular community. You can actually look into large-scale modules. Um, but we like to look at the actually very small motifs. So that means it could be formed by a few cells with a particular cell types combinations. For example, let's say we have three cell types that form a triangle. If this triangle is enriched in many places and across many samples, and this may be meaningful and may be related to cell function or disease. Um, so we actually use a graph network approach to study this problem. We call the TrimNN. So here we start from spatial transcriptome, and then each node represents one cell. And then we use this so-called delineate triangulation to build connections between cells. And then we index all possible motifs for these cell types. But in order to search each motif against the graph, it's actually very time consuming. So we use this so-called graph isomorphic network approach to do the search. That is, the input is a particular motif plus a big graph derived from spatial transcriptome. Then this graph isomorphic network can instantly tell whether the graph contains such a motif. Then we can identify whether a motif is enriched in the data. And then we see if it's enriched, whether they have different survival curves, etc. So here we actually did a simulation. We found the graph network approach is actually almost perfect comparing to brute force search. And then we do see some interesting patterns. For example, this pattern is identified from Alzheimer's disease mouse. Then these four cell types form this kind of a motif. It's overrepresented. So we believe such a motifs could be used as a network marker to differentiate between cancer and the normal, and also various cancer types, or it may even be able to predict immunotherapy response effects, etc. So here we show the one example. So we identified a few enriched motifs. For example, it could be two cell types, three cell types, or four cell types. And then you see this is between cancer and between control. When you have only two cell types, actually the survival curve enrichment is not very obvious. But when you go to four cell types like this, so this motif actually is very clear. And then even within cancer, between more enriched and less enriched, you see the survival curves are dramatically different. So this indicate such kind of a cell type, yes, it's a structural motif, may be a good indicator for disease or survival, etc. Of course, you don't have to really use graph neural network approach or machine learning method. In some cases, just a simple statistical method would work even better than machine learning method. For example, this problem we actually worked on is so-called spatial variable genes. This is actually very similar to differentially variable genes in typical bulk RNA-seq data, where you can see fold change versus a log p-value. Some genes actually differentially expressed. So the idea of spatial variable genes is that on like 2D space, you see the gene expression of a single gene. It may form a particular pattern. That means it's different from random. Then this may be important for this development, cell development of functions. So we actually develop a very simple method, but it works really well. We call it dimension agnostic and granularity-based spatial variable gene identification. So it's based on a very simple idea. That is, if a gene expression has a certain pattern, let's say we use this dog as a pattern, versus it's random, then you see that when you reduce the resolution, this pattern is still there. If it's almost random, when you reduce the resolution, it's pretty flat. So we calculate the standard deviation of a patch, scanning on a picture. So we use a small patch to do it, a large patch to do it, and then we basically get the ratio between large patch versus small patch. So if a gene is a spatial variable gene, then the large patch will maintain pretty significant standard deviation. But the small patch, if it's if without spatial variable pattern, then the ratio actually will be very small because between this and this, there's not much difference. So the variance actually is still random. So by studying this kind of pattern, you can identify spatial variable genes. So such a simple method actually draw a lot of attention because in some third-party assessment of spatial variable gene identification, we were ranked #1. And also our method can really handle very large number of cells, like up to million cells, and can work on desktop, finish within 1 hour. So that's why we got more than 20,000 download just for this tool. So you can give it a try if you work on spatial variable gene identification. Yes, next, I just want to talk about the prompt large single cell RNA-Seq models. So we, of course, use a lot of large language models like ChatGPT and others. So in large language model, what you have that you have these sentences, and then you train a model by deleting, let's say, a word, and then try to repredict the word. So by doing this guessing games many, many times, then you can get a very good vector representation of the language. So that's the basis of ChatGPT and others. And single cell, you can use similar idea. That is, you can consider one cell as a sentence, gene name as vocabulary. So each cell, if you sort the genes by gene expression, it forms a unique sequence. So you have 1,000 cells, you basically have 1,000 sentences. Or the other way to do it that each gene expression value, you can discretize it. Basically, in machine learning, called tokenize the expression value into bins and treat bins as a word. And then you can do the same thing like this. So people have developed many single cell RNA-Seq foundation models using this idea. That is that they collect millions of cells and then they try to predict gene expression value after you delete it. And then you do this many, many times, you get a good foundation model. And then you can use this model to do various downstream tasks. Because it is trained on large cohort of a single cell data, you naturally get background information. So you can get a much better signal to noise ratio. You can correct batch effects to a certain extent. But we and others found that if you use this model directly, it has some limitations. For example, if you use the model for a new task, a new data, it may not work very well. So it is actually questionable whether these single cell large models can be called foundation models, because the true foundation models should have this so-called emerging intelligence. That is, when you use the model for a completely new task, it should work. But so far, single cell foundation models, none of them have this capacity. Um, but we believe that it is still very useful. Um, it requires some special adaptation or some prompts for them to work better. So let's see how this model work. And here you see that if you do the embedding, let's say without any foundation models, you project cells into two-dimensional space. So each dot represents a cell, each color represents a cell type. So different cell types really mix together. But by using different kind of single cell models, and then you do this projection, you start to see different cell types grouped together. And then not only you can do this visualization, you can also measure by all kind of metrics to measure how good uh, the same cell type can cluster together. And all the models we tried, scGPT still performs the best in this regard. You see that it separates different cell types pretty well. But if you fine-tune it, we see uh, quite some problems. This is actually related to this machine learning problem. That is, if you have a big model and then you use small data to fine-tune it, it will have this so-called catastrophic forgetting effects. That is, if you have let's say, this two group of data, this is one group of data, this is another group of data, you have, let's say, two classes. If you use some small data to fine-tune it, you can arbitrarily draw a boundary for this small data. And then you tend to forget the previous data. So here you draw it, then the previous knowledge you learned would be forgotten. But if you use some simple model, that means you only modify part of the model and keep most of the model intact, then it tends to keep the old knowledge. And then you can use new data to refine the model, and it works for both the new data and also the old knowledge. So this is actually the basics of our approach. We actually just published this paper this year in Nature Machine Intelligence, and also we make it open source. We call this SCPEFT. PEFT stands for Parameter Efficient Fine-Tuning Method. So the basic idea is that you have this foundation model for single cell. Typically in the foundation model, you tokenize genes, and then you get a gene embedding encoder, and then you do projection for various downstream tasks. So here, when we use this single cell foundation model developed by others, we simply add some prompts to the model. For example, it could be token adapter. That is, for input token, you have this small unit to modify it. Or you can use a so-called prefix adapter. That means that in the gene embedding, you fix most of it, but you add a little bit of prefix. So when you train a model, you only tune this part. Or you have this LoRa, that means you use a small component, so-called low rank adaptation module. Or you can use this encoder adaptation module. So all of these basically insert a small component. And then when you train the model, you keep the foundation model intact. You only tune this small component. By doing that, you avoid catastrophic forgetting, and then you can really work with small data. And then this allows us to handle a wide range of downstream tasks. That is, you can do cross-species adaptation. You can find this condition-specific gene significance. You can do unsupervised learning to do better clustering. You can do cell type annotation. You can do perturbation analysis. You can correct batch effect. So just show some simple examples. So here is a workflow for cell type identification. So again, for this single cell foundation model, you have this big block, and then you fix it, and then you add one of these modules. And then when you annotate cells, you have some training data. That is, some cells, you have both RNA-seq data and also the cell types. Then you just use this label data and one of the prompt to train a model. And then for new cells with gene expression data, then you can just apply the model to new cells to get cell type annotation. So we found that using this approach, actually this is SC-PAFT approach, it actually works a lot better than fine-tune method in terms of cell type and identification accuracy. And then it's also a lot better than the native method. It's actually cross-board for different kind of large single-cell models, from SC-BIRD, GeneFormer, to SC-GPT. So again, it really demonstrated this prompt approach can resolve this catastrophic forgetting to a large extent. So here we use other cells to do prompt adjustment, and then we get some new cells to see how it actually separate themselves according to different cell types labeled by different colors. So these are a real cell representation, actually. We have 4 cell types. So if you use a foundation model like scGPT, then you actually get some better representation. So different cell types tend to separate better than the raw data. But if you use fine-tuning, you can separate better, but still some cell types not separate very well. If you use this PAFT approach, all the cell types can be separated very cleanly. Another aspect is that we use this differential attention analysis for genes using this SC-PAFT. So the idea that when you have like say 2 measurements, for example, let's say for plants, you have the basal condition. You can have let's say drought or stress condition. So can you find the differential expressed genes? Of course, in the bulk data, you simply just do the statistical analysis. But in single cell, you can do different approaches. You can get different cell types. And then for same cell types, you can compare between the case and the control. But that actually has a limited power. So we use this differential attention approach. We found it actually can gain more analysis power. So in this case, we still use this SC-PAFT approach on top of single cell foundation models. And then we'll build attention. So that is between all the genes and against all the genes, you can get this attention matrix. And then you can also get the so-called CRS classification label that basically represent the global property. And then you can see each gene in target and in control, how much attention you can get. Then you can do this comparison. So this actually is a group level comparison. And then we do see it has a little bit more power. So here you see that we have different cell types. CD for naive, etc. And then for each cell type, we know the marker genes. And then we see that in some cases, of course, CD4 TCM, the marker genes are highly expressed just simply by expression for these cell types against all the other cell types. And then this is actually consistent with our attention score. But in quite some other cases, you see that some of the genes are not differentially expressed using statistical methods, even though people know they are marker genes for this cell type. So this is a CD4 SG15. For example, this gene is not really highly differentially expressed. But using our attention approach, we can really mark them. In many cases, our IC-PEFT approach can mark those genes better than native functional model or fine-tuned functional model. And then here, in another case, we see the marker genes distribution in its attention score. So if you see the native scGBT, so those marker genes, yeah, many of them are outstanding from the distribution, but some fall into the distribution. And then fine-tune approach, the same. But using PEFT approach, pretty much all the known marker genes really are outstanding from the background distribution. And then this approach actually also very fast. So if you look at the scGBT in terms of number of parameters, of course, the PEFT approach, different PEFT approach, only represent a very tiny bit of parameters to be tuned compared to the foundation model itself. So it's more efficient. Of course, the memory usage is not that dramatic because in order to use the PEFT approach, you need to load the foundation model also. So it doesn't save a lot, but still save quite some memory. And then it also actually works well for small data. So here shows downsampling one data set. So even you reduce the data set to 40%, even to 20%, the accuracy can pretty much retain. Of course, the data, if it's too small, it doesn't work. And then you see the PEFT approach, it also requires fewer steps or fewer epochs in training to reach good accuracy. So here I also show some other applications. That is, for cross species, you actually can use orthologs and then to build on top of the PEFT to fine-tune the model. That is, you only tune the PEFT module. And then by doing that, we actually can transfer model from human to other species because pretty much all the major foundation models only apply to human. So here you see that we can even go to C. elegans, which is pretty far from human, and it also show significant power. And for cell type identification, it works better than single R and some other methods. You can also use this approach for unsupervised learning. That is, if you just adapt your model to so-called self-supervised learning. That is, you have this gene expression for single cell, then you repredict single cell without label. Then you build a better representation, and use this representation, you can do clustering, and we see that this clustering actually works better than the original single cell model. For example, here you see that when we do this clustering for this data set, it maps to ground truth label better than the native scGBT, fine-tuned scGBT. In some region, we cannot map. For example, this B cell in our clustering, it shows two clusters. And then we see the gene expression patterns of two clusters. They're actually quite different. And then these genes, if you do gene enrichment test, their pathways are also different. So by collaborating with our experimental experts, so it actually annotated this class 3 and 7. They represent more detailed cell subtypes. So class 7 is actually developed B cells, and class 3 is developed B cells. Class 7 is activated or memory B cells. So it actually gives us a lot of informative annotation. So finally, I just want to talk about the single cell foundation model for plants because it's a plant audience. This actually this work is mostly led by Trupti Joshi. Yeah, she actually worked with me for many years, but now she actually is a full professor and actually dean at Marshall University. It's also in collaboration with Mark Liebold, an important player in this plant cell atlas. In fact, this data we obtained from plant atlas for different plants. So you see here is a spread of the data for Arabidopsis, your corn, soybean, rice, etc. Even though plant single cell data are a lot less than animal data, but it is actually sufficient to build some large models. So here we developed this IC plant annotate for cell type annotation. So input, you start from single cell RNA-Seq data, you build a gene by cell matrix, and then you enter into a foundation model. Then we do supervised learning for cell types. And then we use this model to annotate new single cell data. So this actually is a new transformer-based model. It's probably the first high-performance model for plant single-cell transcriptome. So this is the architecture of the model. Yeah, so we first of all get the single-cell RNA-seq data, and then we select highly variable genes, and then we put into a transformer, and then we predict cell type. We first do the supervised learning, and then we use the model to predict cell types. And this is the kind of the architecture. It is mostly the transformer architecture. And then we found that this cell type prediction using this model actually works much better than other methods. For example, you can use a SCAMP or IC deep cluster or single R. So our IC plant annotate achieved much higher accuracy. So this actually is the apple-to-apple comparison. That is, we use exact same training data, and then we actually work on exact same independent test data. So the single R, of course, works very well in human study, but in plants, it probably didn't have a lot of training data. So that's why we see significant improvement over single R. And this problem sounds pretty straightforward. I mean, the single cell type annotation, but people keep actually identifying new cell types, even given cell types. Yeah, some collaborators, they really like to see more detailed subtypes. So it is still a very useful method. And you see that by using our method, we can really separate different cell types very cleanly on a t-SNE map. And then for this, this is a website address. So here you actually can upload the single cell data in any format, and then it can really give you annotation of cell type. Um, and then it also can do this so-called case and control comparison. It has also a lot of visualization capacity. So in summary, I showed 3 use cases to illustrate how powerful deep learning foundation models can help single cell data analysis. So in the case of graph networks, you see that it really learns a low dimensional embedding of single cell data that would allow you to capture intrinsic cell relationships through this topological information propagation. And then a model scGNN can actually works really well for single cell RNA-Seq data. We have this method called the recept for spatial transcriptome data, and then deep maps for integrating different modality of single cell data, and then network motif identification can help understand tissue functions. And then with this prompt-based approach, you really extend single cell foundation models to many different applications more effectively. And we provide rich libraries for this prompt approach because different problems really have different prompt techniques that works best. Unfortunately, we really do not know the design principle in advance. So most cases, we have to try many kinds of prompts in training data to see which one works best. Then we use that approach for prompts. And then we have this IC plant annotate for plant-specific large single-cell model. So here are the major developers of the method. And here are our collaborators. Of course, with the plant single-cell atlas, we mainly work with Mark Lieboard. And then Juexing actually, so he led work for scGNN. And then now he's assistant professor in Indiana University. The most of the single cell work I actually collaborate with Dr. Qing Ma at Ohio State University. Yeah, so here the support information. And we, of course, also like to acknowledge National Computational Resources, NIR, and NVIDIA for providing a lot of GPU hours for us. Thank you for your attention.
Thank you very much for this really exciting and comprehensive and clear presentation. So I'd like to ask audience if you have any questions about the presentation, please put your questions in the chat, or you can just raise your hand and we can let you to ask your question online. I think while the audience are, do we have any hands raised? So while the audience are contemplating about their questions, I can start with quite a few. But
Okay.
so one of the things about the prompting, because we are very familiar with the prompting with ChatGPT, which is basically natural language. But then my question is basically what type of prompting are you using for the single cell data? Are you prompting with the gene names, gene list, or the matrix of expression levels? So what type of prompting has been used?
Yeah, that's a good question. Yeah, maybe I didn't explain clearly. So prompts is something applied to this a foundation model or large language model. But there are actually 2 kinds of prompt. One is called the hard prompt, like the text you enter into ChatGPT. So those are instructions. So those prompts, they cannot be modified. It's just one way to give instruction to large language model and guide language model to do its task. The prompt I talk about is so-called soft prompt. That means that you add something on top of a foundation model. But then you use data to tune the parameters of that module of the prompt. So that's why it's called a soft prompt.
Mm-hmm.
By adjusting that module, then you make the overall this model more effective.
Okay, I see. Thank you very much. Any other questions? I think it's mostly thanks for the very good presentation. Yes, we have one from the Q&A. Can you see that or can read it?
Yeah, I can read it. Yeah. So the question is about sharing the presentation. Yeah, I'll be happy to share the presentation. Yeah, maybe I will email the organizers and then yeah, feel free to post anywhere or give it to the audience. Yeah, so yeah, I'll be happy to share.
So there's actually a question in the Q&A session.
Yeah.
So it's from, I don't know how to pronounce the name, Ines Marias. Thank you for the great talk. Would these models also work with single nuclei RNA-seq, as this technology is often used in difficult digest plant tissues? If so, have you noticed a difference between single cell and single nuclei RNA-seq?
Yeah, that's actually a good question. So there is there are some differences. So yeah, I think yeah, let's say in the uh, yeah, so there is I would say there's some systematic batch effect because a single nuclear, the way it collects the data is different from a whole cell. We do see it applies across the board. For example, the using this spatial transcriptome, different platform, different methods, they are quite different. So our model, we do not really separate that. So in scGNN, it's data specific, so it works for any type of data. So you in a single nuclear data, you just apply directly. But using foundation model, I think it will be good to make some adjustment. And so that is you have your own data. So you see that all the cases that I talked about, you have this the general model, and then when you apply to a particular use case, then you use your data to either adapt it to some supervised learning, or you can do this self-supervised learning. That means that you adjust the model a little bit by predicting self, the so-called self-supervised learning. So by doing that, you actually adapt the model for your own particular data. So in this case, it doesn't matter what kind of data you have.
Okay, thank you for the answer. So there's a technical question in the Q&A section. I will read that first. It's from Mingyao Hu. Thank you for the very interesting talk. Could you explain the biological interpretation of differential attention? For example, if a gene is not strongly differentially expressed but shows high differential attention between conditions, what does that tell us?
Yeah, that's an interesting question. Yeah, we actually, the reason we actually work on that because many cases we work on this sort of transcription factors. We know transcription factors, they are highly important, but most transcription factors have a very low expression level. And when you have a low expression level, you do statistical analysis. It's actually the power would be insufficient. So many cases, when we work with collaborators, we have to look into transcription factors specifically because a lot of like simple ANOVA approach wouldn't be able to pick up the transcription factors, even though we know they're important. So that's the kind of the rationale we use the attention approach. So attention approach is a little bit different from statistical approach. That is, it's not just based on distribution, but rather based on so-called influence. That means gene, how much influence to the other, or to the downstream effect to other genes, etc. So by doing that, you'll be able to pick up the signal. And then this only works when you have a lot of data, because if you don't have a lot of data, you won't be able to do it. Let's say in bulk RNA-seq, if you only have three measurements in case, three measurements in control, you won't be able to do that. But the advantage of a single cell, you have thousands, even millions of cells. So the data, the information is actually there. Then by using the deep learning method, we actually can aggregate a lot of small signals and then really predict the impact of a gene to, let's say, to case and control. And second of all, it's actually more global. That means this impact is not just a one gene, a one-to-one mapping, but rather a gene is a part of a big network. So attention approach is trying to capture the global property. So that's why it has a more enriched power to find those kind of genes. And then those genes could be lowly expressed, but still visible through the attention approach.
Thank you for your answer. So I think there was a question put back, but I'm going to continue reading the one still opening. Professor Xu, is single-cell seq important to understanding plant nutrition and nutrient management in crops of economic importance? This is from Benjamin Kari. So the question is more generic. Can you use single cell to understand plant nutrition and nutrition management in crops?
Yeah, I think yeah, single cell can be used for any problem. Let's say in the nutrition area. So, for example, people study the oil content, protein content, or let's say omega-3 or special kind of nutrients. And many cases, let's say if you design a crop to get let's say specialized nutrient composition, or sometimes people like to have a more lysine in the crop, etc. And single cell could be a really powerful approach to study that. For example, you see at this production level, different cells or how they regulate this. So the point that people already use bulk RNA-seq data to study these problems, but single cell give you much higher resolution, is more informative. So pretty much whatever done by the bulk RNA-seq, nowadays people use this single cell RNA-seq to do it.
Thank you for the answer. And so the next question from Zahor Mir. Hello, such a great talk. I question, if our data has batch effect, will it be normalized using the training model, your models? So the question is about batch effect. Could any of your model be used to correct for batch effects?
Yeah, so batch effect in single cell is actually a pretty big problem. We see that there are many tools to do batch corrections, but they don't correct perfectly. So many cases it perfect to it actually depend on different criterion. It may not correct very well. The foundation model actually correct a little bit better because you really see a lot of data. And then one pressure actually is that we have to try many normalization techniques. Yeah, I didn't have time to talk about that part because that's not related to single cell. So we're developing this framework, kind of a workflow pipe, so people can select more than a dozen normalization approach, and then you can get let's say different results. Some results align your interpretation better, but then that probably is a better way to do normalization. So, so my own experience that a lot of cases, instead of using advanced methods for batch effect, it may be better just try like different kind of let's say how to just do simple normalizing. For example, whether you divide by a certain average or something. There are actually more than a dozen approaches to that. Then you see which one works the best. And then a further step, we are trying to develop an agent approach to do this in a more automatic fashion, but we haven't started that part yet.
Thank you. So many people are interested in using AI agent.
Yeah.
So the next question is, are these adaptable for sequencing-based spatial transcriptomics like Curio Seeker, which is a specific platform for spatial? transcriptome?
Yeah, actually, this spatial transcriptome component, for example, with those spatial variable genes, all those things, they really work on any kind of data. It could be spot level or single cell level. Single cell level could be image based or sequence based, or it could be codex protein based. So any data would work. But the sequencing based in some cases, yeah, you do need to yeah, actually do some special processing because sequencing based, for example, the yeah, some latest methods, their reads is still very sparse. That is, for example, Visium HD, each cell actually pretty sparse comparing to RNA-Seq. So standard operation is that people do sequence-based spatial transcriptome, and then same sample they also do standard single-cell RNA-Seq. So they combine the data in order to address this sparsity of sequence-based spatial transcriptome data.
Thank you. Thank you. Okay. So I think we are almost at time. We have two more questions left. The one is, it is from Fatma Karateki. Is it possible to create a 3D model that predicts the genes of plants? Generally, when making predictions considering external factors, we encounter a stochastic process. This is interesting. So it's about stochastic process and predict 3D model of gene.
Yeah, so I think in animal people are doing that actually.
Okay.
So in plants, I don't see any work, but in animal people are really build so-called virtual cell or digital twins. That is using single cell data, other data, and also incorporate stochastic effects. So they have both biological knowledge graph, and then they also have a differential equation to model the kinetics, dynamics, etc. Yeah, I mean, there are a lot of papers on that. So the goal is that they really want to model a cell kind of a 100%. But I think that goal, I don't see it achievable. But in some special cases, for example, if you model, let's say, drug effect in a very special setting, it really works. So that kind of a specialized digital twin or virtual cell, I do see a good prospect. And this could be extended to plants in the future.
Yes. So we are already at top of the hour. We want to respect Dr. Xu's time. If you have time, we still have more questions coming in. You can keep answering everybody else. Thank you for attending. And I just want to shamelessly advertise, we actually have a workshop at Genentech working on single cell sequencing. My question to Dr. Xu is that, would it be possible for one of your students to come to here or do a live demo on Zoom for how to use the plant single cell annotate, which, just do a demo to show how we can use the platform.
Okay, so yeah, maybe you can send me more information.
Yeah, I will send you an email.
Yeah, okay, yeah, that'll be great. Yeah, thanks.
Okay, thank you, everybody. And we do have more questions. If Dr. Xu, are you able to stay longer?
Yeah, I can stay online. Yeah.
Okay, perfect. So I'm going to read the next one.
Okay.
It's from Mingyao Hu or G, I don't know. You showed a promising cross-species transfer using autologous mapping and PEFT. How sensitive is the model performance to the quality of autologous mapping, especially between more diverse lineages? More broadly, do you think we need to build a separate foundation model for each species, or could one broader plant foundation model work across species with species-specific adaptation modules? That's a very good question. I want to ask that too.
Yeah, that's actually a very interesting question. Yeah, in fact, we are debating ourselves. Yeah, I even talked with ICGPT model, is a lead author, Bo Wang. So I asked him, because he worked a lot on different species, although he doesn't work on plants. So the question, actually, we were talking about whether we should train one model for many species, or we use a human model to transfer to other species, or we use one model data to build a specific model for each species. So those are different strategies. At that time, he thought that I mean, at least at that time, he thought it's better just use the mouse data to train a mouse model, C. elegans data to train a C. elegans model. Um, yeah, so of course so that could be one way to do it. Our prompt approach, we found that at least before that kind of model is available, we can adapt to pretty much any species very quickly and also very efficiently. So we do see some advantage. But in plants, it's a little bit different story. Plants, I think the reason why there are not a lot of model for plants. First of all, it's actually a lot harder problem. And, and second of all, there are much fewer data available. So let's say if you train a model for some plants with very little single cell data, I don't think it is effective because if you don't have, let's say, millions of cells, or at least, in my opinion, 100 cells, you may not be able to get a very effective model. So most most plants won't have that many single cell data. Another challenge that plants, many cases, you have a lot of duplicates. I mean, I would say paralogs of genes. Yeah, actually, sometimes single cell data may not be even to resolve between paralogs. And then vocabulary could be a lot bigger. So we do see for plants, it's a lot harder. So that's why our model focus on limited number of species because we have at least a lot of data for those species. And then we can build a good vocabulary and the gene annotation of those species are relatively more reliable than many other species. So I think this might be the solution for now. Of course, when single cell data are accumulating a lot, it could be different. But so far, I think our approach may be at least practical at this point.
Very insightful answer and very interesting. Yeah. Any other questions from the audience for today's speaker? Oh, we have one more. We're going to from single cell in Arabidopsis root hair, going to getting immune response in root hair and lateral root, but not in the main root. I check it with calcium signaling RS accumulation pathogen, and I'm using is a nectar trough, and I also check the pathogen entry using WGA staining. What will be the best approach to identify the factor that trigger immune response in root hair? I think that's the key of the question. In the specific cell type root hair, how do you identify the factor that trigger immune response?
Yeah, so yeah, our approach actually can help build some networks. So that means cell type specific networks. Yeah, but of course, the machine learning method sometimes hard to use because you need to really get the GPU and run them. So for simple solution, I mean, simple solution, I would suggest that you first of all get single cell data. You run simple tools like Thread to get cell type annotations. And then let's say if you're interested in particular cell type or tissue type, let's say root tip, you just collect all those cells for that cell type, and then you treat it as a bulk data. That means may only represent 10% of all your data, but that's a pure cell type. And then you use the standard bulk RNA-Seq data. For example, there are quite some tools available for bulk RNA-Seq data, gene regulatory network analysis or things like that. So that could be one way to do it. In some other cases, if you can go a little bit more sophisticated, then you should run this so-called cell trajectory. That means you basically can get a trajectory of a development stage. Then your cell could be like in a kind of a one-dimensional space. That means that it's sort of pseudo time. It's almost like you can treat it like a real time. The different stages, you have different cells, and then you can treat it as a kind of a trajectory. And then actually a lot of tools to analyze that kind of trajectory, to build the regulatory networks based on trajectory. So even though those tools are intended for real time, here you only have what's called pseudo time, but it probably can give you some good insight. And the bulk RNA-seq analysis tools, they have been used for many years, like 2 decades. So a lot of tools are very mature, even though they are not intended for single cell data. But I would suggest that you can try that route.
Do we have more questions from the audience? No, no more questions. I think this has been more than 10 minutes, over an hour. Thank you very much for Dr. Xu for the really insightful and presentation and clear presentation and all this question answer to the audience. Thank you very much. Let's a round of applause on Zoom for Dr. Xu.
Okay, yeah. Thank you, Song, for hosting it. Yeah, I'd like to thank the audience for coming to my talk. I really enjoyed the discussion.
Yeah, I enjoyed this talk and discussion as well.
Thank you so much.
Okay, thank you. All right, so I will email you the slide. Yeah.